Okay, this video is going to be about cancer. We'll call it the pH paradigm of cancer. And I got started down this path when I was reading The High Blood Pressure Solution by Richard Moore. He devoted his life, over 40 years, to studying the plasma membrane ion pumps in the context of hypertension. But he said some very interesting things about diabetes, that with high salt diets you get elevated blood insulin levels, okay, because you get chronic increased cytoplasm calcium due to dissipation of the sodium gradient. And I'm going to show you what it all means. But the bottom line is you get elevated insulin levels, and the elevated insulin levels cause pumping of protons into the extracellular space, acidification of the extracellular space. I'll explain more what that all means, but here's a couple quotes. Richard Moore, high insulin levels cause acidification of the extracellular space. And I'm like, gee, that reminds me of studying bacteria physiology. Bacteria pump protons into the extracellular space, and with good reason. Nick Lane, he's a real smart guy, writes a lot about biochemistry of the mitochondria and origin of life, and he says all bacteria want to do is replicate. Bacteria have a proton gradient across their plasma membrane. They pump protons into the periplasmic space, which is the space between the plasma membrane and the cell wall, and it's like an extracellular force field. It's like a proton power force field around the Starship Enterprise, like from the TV show movie Star Trek. Okay, where am I going with all this? I'm trying to teach you about cancer, and I'm trying to teach you to see things in a better way through the metabolic theory of cancer. Because once you get all this stuff, it becomes obvious what you should do for your own health. I'm empowering you intellectually and for your health by understanding these metaphors. And the metaphors make it easier to grasp concepts in biochemistry and pathophysiology. That's why I'm doing it this way. It'll all make sense in a moment. Okay, we talked a lot about sodium. We talked a lot about the plasma membrane, potassium-sodium ATP pump. You're pumping out 3 positive charges for 2 positive charges going in. So that produces an electrical current. This is an electrical generator, this pump. You separate charge across the plasma membrane so you have a voltage. That's a battery. That's what a battery does. It can generate current on demand because it has charge separation of a voltage. Okay, so the charge is carried by the positive ions of the sodium. They want to equalize their gradient of concentration, and they also want to move towards this negative charge, so the sodiums want to come into the cell, and that can be used to pump calcium out. Okay, it can also be used to pump other things out of the cell. Now here's a little more detail about a typical cell, and one of the things you're going to notice is that you can also couple bringing sodium into the cell to the pumping of hydrogen protons outside of the cell. Hydrogen proton is acid. So remember, only the ATP-driven sodium-potassium pump pumps sodium out. Everything else brings sodium in. So this is where the cell has to make energy to make this pump work. Everywhere else, the energy produced by this electrical battery, this gradient, is harvested, used to do work for the cell. But what's most important for our purposes today is this right here. This means natrium, that's sodium in Latin, and then H is for, helium, hydrogen, okay? Sort of named after, just remember hydrogen. All right, and then X is for exchanger. So you bring sodium in, you pump hydrogen protons out. And if you pump a lot of hydrogen protons out here into the extracellular space, you will acidify it. To have more proteins means to acidify. And an acidic pH means a low number for pH, okay? Okay, then I was reading this article. I had remembered what Richard Moore had written. I read a little bit about it, but I got busy with other things. Then I came across this paper, The Effects of Systemic and Local Acidosis on Insulin Resistance and Signaling. So what happens is when insulin binds to the cell, it causes more glycolysis to occur, the burning of glucose for energy. That generates lactate and protons, lactic acid and protons. And then with insulin present, more lactic acid, more protons are pumped out of the cell. You can also pump out lactate, but as far as we care about, what we care about is that more hydrogen protons are pumped out of the cell. I'm like, well, gee, that's interesting. That's nice, but so what? Well, this is actually a big so what. In small amounts, this is normal, okay? But in big amounts, like when you eat a lot of fat and you have a lot of insulin resistance and you get excessive amount of insulin here, despite the fact that you have more trouble getting the glucose, GLUT4 transporters to go to the plasma membrane, you end up pumping out more protons, okay? Remember that. Insulin's mitogenic, causes cell division, but it also causes acidification of the extracellular space. That's going to be very important. Okay, a little bit of detail about it. We don't need to go into it. Here's one paper you can look up if you want to look up papers on it. I'm going to give more lectures on this stuff and go into more detail, but what you need to know is that insulin causes acidification of the extracellular space. Now, this guy with his group, Stephan Reshkin, did some really great work, and he wrote a great paper here. I'm going to show you this paper. And so pHE, the E is for the extracellular space, and that becomes acidified with cancer. So ES is extracellular space. The pH in the extracellular space will get as low as 6.2. If you remember, blood has very strong buffers for pH, and it keeps the pH real tight between 7.35 and 7.45. That's normal blood pH. But the extracellular space does not have that much buffers. So pH can get very low as like around 6.2. And when that happens, it has major effects because simultaneously, when the extracellular space is acidified, the intracellular space is going to be alkalinized, means made more basic. So here's a symbol for intracellular space. pH, which relates to acid scale, and then I is for intracellular. Cytoplasm is, of course, intracellular. You're going to be running more glycolysis. Okay, when you run glycolysis, even though oxygen is present, that's another name for the Warburg effect. The Warburg effect is, lack of oxygen, hypoxia, same thing as lack of oxygen, causing mitochondrial injury, leading to the cell to change and emphasize anaerobic glycolysis. Emphasize glycolysis rather than mitochondrial metabolism with oxygen. That's called oxidative phosphorylation. Okay, typical cancer cell has localized hypoxia at its location, and it runs on more glycolysis. A cancer cell will take up like 100 times more glucose, and that's why cancer cells are positive on PET scans. By the way, what I'm getting towards is the theory of reverse evolution, that cancer represents a reversal of evolution. So evolution tells us the story that single cell organisms like bacteria, also known as prokaryotes, so-called evolved into multicellular organisms that have a nucleus in their cells and that have mitochondria and can run on oxygen metabolism. Okay, so we're going to get to all of that, but the important point is that in cancer, it's like the progress of life has gone backwards, and a cell from a human body is basically turned into like an anaerobic bacteria. And it does relate to the idea of facultative anaerobes, related to the idea that when the world was started by God, there was no oxygen until later on cyanobacteria developed the ability to undergo photosynthesis. And initially the oxygen on the planet was relatively scarce and intermittent. So cells that could run oxygen metabolism had to be able to switch back and forth from oxidative oxygen metabolism to anaerobic metabolism. And therefore, they've got that somewhere in their DNA program, this primitive way of life, anaerobic glycolysis, okay? And the reason this matters is that's what cancer really is. It's like a human cell turning itself into a bacteria cell. And all body cells have the same DNA. It's just a question if they can turn on those old programs. So a facultative anaerobe is like an opportunistic anaerobe. And a cancer cell has turned on those old pathways to run largely on anaerobic glycolysis. Cancer cells will still have a few functional mitochondria, and it depends where they're located in the tumor relative to the periphery of the tumor. So for the cancer cells near the periphery of the tumor, it might have a little more mitochondria and run some oxidative oxygen metabolism. The deeper you get to the center, the more hypoxic it is, and the more anaerobic it's going to have to run. But the important point of this talk is that the pH gets changed by high levels of insulin and this transformation into cancer. Because more protons are pumped out, you get an acidic extracellular space. And this is a characteristic of all cancers. And this is the name of the paper. Here's Stephan Reshkin. He's the main author, the sodium-hydrogen exchanger in the reprogramming of cancer metabolism. Okay, and as far as he's concerned, this is the most important thing to know about cancer. This is the cornerstone of all cancer, and all cancer cells have this. And this involves a reversal of the usual pH gradient in a functional cell. And this is very interesting because the anaerobic situation favors running metabolism out of the cytoplasm with glycolysis, whereas when oxygen is present in a healthy eukaryote human cell, you're going to want to make your energy from the mitochondria. It makes, about 18 times as much energy per ATP as anaerobic glycolysis does. Okay, I'm going to show you some more pictures. This is going to get more interesting. I had to give you that little warm-up so we get to the cool stuff. Now here's sort of the cool stuff. This is like the academic orgasm of this talk. And here it is, that you have a pH gradient between the inside of a cell and the outside of the cell. And when you acidify the outside of a cell, then you're going to run on glycolysis, okay? When you acidify the inside of a cell, it's the opposite. So the important point is that pHI means pH intracellular. It's like a thermostat for the cell, okay? Under normal conditions, you actually have a slightly acidic pH inside of a cell, and that actually favors oxygen metabolism to run your mitochondria. That's actually a good thing under normal conditions. I'm talking about intracellular. What really matters, though, as far as like what goes into your blood and your diet, is what's happening to the extracellular pH. Now, when you have anaerobic conditions, you will get acidification of the extracellular pH, and that's going to lead to anaerobic metabolism running glycolysis inside the cell. Okay, but what I'm trying to say here is oxygen conditions of healthy oxygenation lead to a relatively alkaline extracellular pH, and the cell makes energy from oxygen, and everything is good. It's happy. All right. And this idea of a pH gradient going from intracellular to extracellular space, and the more severe it is, the more acidic the extracellular space is relative to the intracellular, the more aggressive the cancer, the more likely it's going to grow, metastasize, and kill the patient. So this is kind of interesting. Okay. And here's the same diagram written bigger. Okay. So you don't want your extracellular space acidified. Now another question comes up, well, gee, ionic composition is relatively similar from the blood to the extracellular space, and that's because there's osmotic gradients where the blood and the extracellular space are in relative continuity. However, the extracellular space is different than the blood, very much so when it comes to pH. Even though it's very similar for ionic composition, it's actually quite different for pH. And the reason is the blood has to control pH very precisely. So it's got a very strong buffering system, like with bicarbonate. And you also get some buffering capacity from the hemoglobin and from the other protein, the free-floating plasma protein, the albumin. But in the extracellular space, you do not have good pH buffering. So if a cell starts pumping out a lot of protons like a cancer cell will, also as occurs from a lot of cells in the presence of high insulin levels, you can get acidification, significant acidification of the extracellular space. All right, this is an important point. This guy Reshkin actually thinks it's the most important thing to know about cancer. What I'm emphasizing in this talk is the fact that abnormally high levels of insulin, as occur with high-fat diets, they cause tissue hypoxia. We know that from Dr. McDougall's lecture, the work of Roy Swank and others. You get tissue hypoxia. Well, and what I'm trying to say is tissue hypoxia and high-fat diets and high-sodium diets and insulin resistance and extracellular acidification, they all go together. They're all related to each other. And they all increase the likelihood of cancer and making the cancer more aggressive. Okay? So here's a little more, and here's some quotes from Steve Reshkin, sort of extracted from his article. This is the pH-centric paradigm of cancer. So cancer based on pH of a cell. The earliest step in metabolic programming of a cell is the reversal of the pH gradient from intracellular to extracellular, with the key point being that in cancer metabolism you get high pumping of protons into the extracellular space, causing acidification of the extracellular space. And this creates a vicious cycle. The excess glycolysis keeps putting more protons into the cell, and they pump them out quickly. They upregulate, make more plasma membrane proton transporters to get those to get those protons out of the cytoplasm as fast as they can. And this will then have the ability to cause the cell to make more DNA. It goes into synthetic mode. I don't know exactly why that happens, but I do know it happens. And it gets the cell to push towards replication. Again, the cell's basically transforming itself from a sophisticated modern human eukaryote cell into an anaerobic bacteria. The tumor microenvironment is characterized by hypoxia, poor blood supply, limited nutrient supply of glucose and oxygen, and acidic extracellular space. The acidic extracellular space, this is pH extracellular, promotes production of more collagen in the extracellular space. That's also bad. An alkaline pH promotes removal of collagen from the extracellular space. And can you also see how that relates to atherosclerosis? Because it does. With an atherosclerotic situation, the tissue hypoxia can become a vicious cycle, like in diabetes and hypertension, where more collagen is made in the extracellular space, which is going to cause more hypoxia of the tissues. You got everything going in the wrong direction here. Okay, and here's a little bit like what I'm getting at. Here would be a normal capillary. You got red blood cells passing through a capillary. The little spindle-shaped cells in the background are the lining cells in the wall of the capillary called endothelial cells. The yellow right here is the capillary basement membrane. The green are the vascular smooth muscle cells. The blue circles are the oxygen going to the cell here, the neuron, the brain cell. Well, with chronic hypertension, you get replication, thickening, increased size, hypertrophy, and replication, hyperplasia of these smooth muscle cells, vascular smooth muscle cells. You also get glycation of the capillary basement membrane and thickening of the capillary basement membrane. You'll also get, as I mentioned a moment ago, more protons pumped into the extracellular space, and that's going to cause more collagen to form here to thicken this arterial wall, like arterial sclerosis. Collagen is kind of like analogous to fibrosis, to sclerosis. And the thicker this capillary wall gets, arterial wall gets, the less oxygen is going to be delivered to the cell. And that's bad, because the less oxygen the cell gets, the more likely it's going to be injured, and have a Warburg effect, injure the mitochondria and switch to anaerobic metabolism. So you can see the vicious cycle that's going on here. And then we talked, I gave a previous lecture yesterday, and this is from the illustration by Guido Hegesy. It's a fair use illustration. He's a medical illustrator. Here's his website, hegesy.de. I think that means German DE. So anyway, it's a beautiful illustration here, and he shows under normal oxidation conditions with widely available good oxygen, the HIF, hypoxia-inducible factor, alpha subunit, just gets degraded. And so the body makes it just in case hypoxia occurs, but degrades it. Basically you stick it with ubiquination stickers, which are like garbage eat me stickers, and it gets chopped up in the proteasome, which is like a wood chipper. And so these amino acids get recycled. All right, so it's obviously pretty important for a cell to know if there's hypoxia or not. When there is hypoxia, lack of oxygen, now these oxygens aren't available. The initial step in getting this process going was hydroxylation of the HIF alpha subunit. But there's no oxygen to hydroxylate it here. So now instead of being destroyed down this pathway, the hydroxy-inducible factor will travel to the cell nucleus, where it'll combine with a beta subunit of hydroxy-inducible factor. When the two of them are combined together, they then become a transcription factor, meaning they control transcription of the DNA. They bind to the DNA at something called HRE, hypoxia response element, and they cause production of all these enzymes to run related pathways for hypoxia, like more glycolysis enzymes. And then you're also going to make more blood vessels, angiogenesis. Okay, so what I'm trying to make the point of is hypoxia and high insulin levels, all this stuff goes together. Also, like I said in other lectures, in the Western world, most of the patients all have the same disease. They start out with high-fat diets and high-sodium diets, and that causes hypertension and prediabetes and diabetes, and that causes atherosclerosis, and all of this causes tissue hypoxia. And they have, in the type 2 diabetes phase, before they destroy their pancreas, they have high insulin levels, and that has a anabolic, proliferative effect, a mitogenic effect, okay? And it all ends up in early death, cancer, and these other diseases, heart attacks, strokes. But what I wanted to show you is, and now I apologize a little bit for the metaphor I'm going to give you. It's a little bit harsh, but I want you to get the point, because I can tell you sometimes I'm sad as a doctor. I see one after another train wreck patient. They never get better. They just spiral into the drain. They die and they suffer miserably. And most of them, by the time I see them, they're already, over 55 or 60. They're kind of cognitively slow, fat, sick, and stupid. And I often don't have much time to talk to them. But I'm trying to help you to see this metaphor so you'll understand. You have a chance to reverse this, to prevent this. And these are some pictures from the movie Gone with the Wind. And I love that movie. It's one of the greatest movies of all time. And Clark Gable as Rhett Butler was fantastic. He was magnificent, okay? And this was his daughter, Bonnie Butler, okay? And he loved his daughter very much. He took her riding on the horse with him. He taught her how to ride her little pony. Then he got a bigger pony. And he loved her very much. He loved her more than anything in the world. I loved my mother more than anything in the world, okay? And Bonnie was kind of a feisty kid, and she grew up, and it was good that she knew how to ride a horse. Back in those days, everybody had to know how to ride a horse. But Rhett Butler made a big mistake. He taught Bonnie Butler how to jump over jumps with her horse under his supervision. And he should not have done that because she's too young to have maturity to make good judgment about that. And she jumped over a jump one day, and she died. And I'm telling you, Gone with the Wind was such a sad movie. And that was one of the saddest moments in the whole movie, was when Bonnie Butler died because she jumped her horse. And went over a jump she wasn't ready for, and the horse fell. And it reminded me of this painting by Benjamin West, Death on a Pale Horse, basically galloping downhill to death. And I kind of feel like, I see this all day long, every day in the Western world. All Western hospitals are the same. It's been this way my entire career in all Western hospitals. The patients have dietary diseases, but nobody in the conventional medical system knows anything about dietary disease, so they always treat it with drugs and surgery, and the patients don't get better. And they have very predictable ends, myocardial infarction and cancer. What I'm saying is, it all goes in the same direction. Obesity, hypertension, diabetes, autoimmune disease, atherosclerosis like coronary artery disease, cerebral vascular disease, stroke, heart attack, AFib, congestive heart failure, and cancer. They're all variations on the same pathway. They're like box variations on a theme, a fugue, okay? And this is how it ends. It ends bad, and there's only one way to reverse it. Go low fat, low sodium, vegan, okay? It's pretty obvious, so you don't get all this stuff going. And it also shows what a joke, all that paleo, keto, carnivore. How are you going to win with meat when it's high in fat, especially sat fat, which causes insulin resistance, which causes obesity, okay, and it also causes hypertension, and then it elevates insulin levels, which is mitogenic and leads you into extracellular acidification of the extracellular space, which is, again, carcinogenic. You're not going to win with it. And so we know the answer. McDougall figured out the answer. All I'm doing is just, pointing out some of the biochemical reasons why it's true. So anyways, I hope that was helpful.